Good day, everyone. We have a great, exciting webinar today, and it's going to be about 3D iPSC culture, and it's using our RocketCell 3D iPSC growth kit, an all-in-one Xeno-Free platform. I am Christopher Oak from TheWell Bioscience, and I'll be your moderator, and also alongside for our speaker today, it will be Dr. Rick Cohen. A few items we're starting during this webinar. You can utilize any questions you have, and we will collect and present them during the Q&A session. Also, the webinar will be recorded, which can be accessed later for your convenience. So let's get started. It's my pleasure to introduce today's speaker, Dr. Rick Cohen. Dr. Rick Cohen is a stem cell biologist with more than 2 decades of experience in stem cell research, iPSC technologies, and advanced cell culture systems. He received his doctorate in pharmacology and therapeutics from McGill University and completed his postdoctoral training in neurogenics at the NIH, NIMBS. Over the course of his career, Dr. Cohen has established standardized stem cell culture protocols, developed viral and non-integrating methods for iPSC generation, and engineered functional differentiation systems across multiple lineages. He has trained over 100 scientists in human stem cell technologies and has contributed extensively to stem cell biology, including work on iPSC expansion, neuronal differentiation, and scalable cultural systems. His expertise spans both foundational stem cell biology and translational applications, with a strong focus on developing reproducible and physiologically relevant culture platforms. Please welcome Dr. Cohen.
Thanks, Chris, for that nice introduction. So today we're going to hear about 3D iPSC culture using our RocketCell 3D iPS Xeno-Free Complete Growth Kit. This is an all-in-one Xeno-Free kit with optimized matrix, medium, and other reagents for 3D iPSC expansion. So before we start, why culture iPSCs in 3D? One, it orchestrating complex differentiation states. We use something like the germ layer interaction. Previously, with embryonic stem cells, people developed embryoid bodies or teratoma formation. So it's very similar. The 3D environment facilitates the co-emergence of endoderm, mesoderm, and ectoderm layers, allowing for crosstalk that actually mimics real embryonic development in vivo. Tissue organization. So in 2Ds, iPSCs lack the spatial cues to organize into complex structures, whereas in 3D, they undergo self-organization, forming polarized and layered tissues like the retina, the brain cortex, or intestinal lining. 2, superior maturation and functionality. Adult-like phenotypes. iPSCs in 2D often remain immature in nature. So for example, cardiomyocytes generated from iPSCs don't beat as strongly, whereas in 3D cultures, it provides the mechanical tensions and electrical couplings necessary for the cells to mature into adult-like functional tissues. B, metabolic realism. 3D iPSC models develop realistic metabolic pathways, making them far superior for toxology screening and modeling complex disease like Alzheimer's or type 2 diabetes. Scaling for clinical applications, the volumetric efficiency. 2D cultures are very limited by the surface area of the plastic substrate, whereas 3D static or suspension cultures utilize the entire volume of a vessel. So this allows for the production of billions of cells required for cell-based therapies. And lastly, homogeneity. Automated 3D bioreactors provide a controlled environment that regulate pH, O2, and CO2 that reduces the edge effects, common variability seen in manual 2D plate-based cultures. So what is RocketCell 3D Xeno-Free Complete Growth Kit? It's a combination of the Vitrogel stem, Xeno-Free supplement, suspension media, basal media, and a RocketCell cell viability enhancer. So how does RocketCell 3D iPSC Xeno-Free Complete Growth Kit help you accomplish your 3D goals? Firstly, there are many challenges in setting up all the steps needed for successful 3D iPS cell culture. And no one likes to waste time, and everyone likes reproducibility. The RocketCell 3D iPSC Xeno-Free Complete Growth Kit solves these challenges. Let's see how. lack of complete integrated system, the researchers often rely on matrices, media, and supplements from various sources, creating inconsistency and workflow complexity. Here, we have an all-in-one complete system, fully defined Xeno-Free kit combining 3D hydrogel, growth medium for seamless pluripotent stem cell culture. For single-cell survival and post-passage viability, dissociated iPSCs often lose viability when seeded as single cells or small colonies. Improve cell survival and growth. Here we supply a reagent that supports both single cell and small colony seeding, maintaining high post-passaging viability and consistent colony formation. Labor-intensive and high-maintenance culture workflow requires frequent feedings, weekend maintenance, limits flexibility and throughput. And here we have a weekend-free workflow and also a possible alternate day feeding schedule, which minimizes handling and supports consistent long-term cultures. Inconsistent performance in undefined materials. Animal-derived or poorly defined components introduce variability and compromise reproducibility. Here we present a system with Xeno-Free and chemically defined composition, ensuring high reproducibility, stability, and regulatory compliance for advanced research and possible translational use. Limited scalability and inefficient use of resources. Traditional systems have low seeding density and high media consumption, limiting expansion. With this system, you have a high-density 3D culture capability. The 3D hydrogel matrix supports up to 10 times higher cell densities than 2D culture, maximizing expansion efficiency. And lastly, others require special handling and not room temperature stable, requires a pre-coating or steps to maintain at cold temperatures, complicating setups, especially in the high throughput format. Here, it's ready to use and room temperature stable, operates without pre-coating or thawing steps, simplifying setup and improving ease of use. So if you're not convinced already to go to 3D, you're not ready, we can also help you achieve robust and optimal Xeno-Free results in 2D. You can get used to our products. The RocketCell iPSC Xeno-Free growth medium is available as a basal medium and a supplement. And this will help you bridge from 2D to 3D. So you can check out our VitroPrime 3D culture vessels, the RocketCell Xeno-Free culture media, and also our CytoGrow vitronectin as a coating. The figure below here shows iPSCs after 4 days in culture on our VitroPrime dish in phase and in red fluorescence. This will give you an idea that we're going to be using this cell line later on for imaging purposes. And bridging the gap between 2D and 3D, you can see here, these cells are developing in an IncuSite S3. So we put the cells in the IncuSite after we passage them on Thursday. This is our 7-day workflow. We double feed on Friday. We have our weekend free, come back on a Monday, don't feed on Tuesday, feed on Wednesday, and passage on a Thursday. And that results in a monolayer of iPS cells. So our 2D system is quite robust, but let's see how to go to 3D. Before we get started, I just wanted to introduce our new VitroPrime 3D culture and imaging plate. They come in 3 flavors: 96 well, high throughput compatible, also compatible with automated liquid handling machines, the 24 well dish for routine assays, immunostaining, small scale grow up, and the 6 well, which we use for production, scale up in static 3D, and also a really important point to produce enough material as a launch point for differentiation. We can also do that in the other formats, but it's more compatible with a larger scale. And these 2 plates I'll use in the webinar and refer to them. If you need more information, you can scan the QR code. So let's look at the data overview I'll be presenting today. Thawing into 3D. This is important because you can use your existing frozen stocks without any adaptation. You can move from 2D to 3D, and that's really the easiest method to get started. With your existing live cultures, you can move to and from 3D. So if you have cultures in 3D and want to move them back to 2D, you can do that too. Subculturing 3D comes in when you want to look at some downstream assays. and also differentiation. And we're going to come back to subculturing 3D a few times in this talk. Some examples of 3D assays we'll talk about called cell counts, metabolic dye, and this is important to give you an idea that you can use your current 2D assays and they will work in the hydrogel with our system. Imaging applications, a live dead assay, important ones that people use all the time, imaging fluorescent proteins, and also doing live and fixed immunostaining with directly labeled antibodies and second step antibodies. This is important to understand that all of our hydrogels, not only the one I'm presenting today, they're optically clear. Also, antibodies and small molecules can penetrate throughout the system, and the hydrogel stands up to processing, especially when using our new VitroPrime 3D culture and imaging plates. Lastly, we'll discuss differentiation in 3D. I'll show you some examples of neural differentiation using a simple two-step protocol. And this is going to be associated with the emergence of 2 new kits coming out very soon, and also recovery into a new hydrogel type. And this is going to circle back to subculturing. It's important. So here it's important to understand that biological systems, for us, are 3D systems. We're not 2D systems. And this kit is really only part of a much larger launchpad empowering you to explore and discover in a more physiological, relevant manner. So let's talk about thawing iPS cells directly into 3D cell culture workflow. So you recover your ample from the liquid nitrogen tank, defrost it either in a water bath, or here we're using an automated tube defroster. Dilute that into 9 mLs of RocketCell iPSC Xeno-Free media with 1x RocketCell viability enhancer. Count the cells, so you can remove an aliquot and count them, and then triage what you need aside, or collect all of them and just use all of them and plate an entire dish if you're starting with millions of cells. So you would resuspend your pellet in suspension media, so we include that in the kit, and you would add 3 times the amount of RocketCell cell viability enhancer, so that will give you 1 volume. In 1 well of a 24-well dish, we recommend starting with anywhere between 50,000 and 150,000 cells. We stick with 100,000 as an advice, and that will come in at 100 microliters. So generally, if you resuspend your cells at 1 million cells per mil, it's compatible with this system. And then you add 2 volumes of Vitrogel stem. And basically for all other techniques, this is what you do. 1 volume of your cells, 2 volumes of your Vitrogel stem or other Vitrogels. Immediately mix 10 times gently. And I can't stress this more. When you mix any type of cell, especially iPS cells, you don't want to do it violently because you're going to kill them with shear force. So use caution, but do mix these very thoroughly 10 times. If you have enough cells, you divide the mixture into different wells of the culture vessel, cover it, and set it aside for the golden 15 minutes. And most all of our hydrogels, that's the time it needs to polymerize. Now, everything here is happening at room temperature. You don't have to be worried about keeping anything cold or heating anything up. Everything goes room temperature. After the 15 minutes, you gently overlay equal volume. So per well, we're talking 100 microliters of cells, 200 of gel, 300. You overlay 300 microliters minimum. You can do more. And that will contain a 1x RocketCell viability enhancer. I always recommend to observe it to see how it turned out, and then put it in an incubator. I'm going to present some images later using the IncuSite S3. If you use an IncuSite or similar, you can put it directly in there. If we were using a 2D plating, I've tried putting it in the IncuSite right away, but the vibrations cause problems with plating, and you can't do that. So with a 3D system, you can observe it immediately. And here's some data. The growth of iPSCs recovered from cryopreservation, and these are thawing acutase-dissociated frozen cells in the 3D environment. This is at day 0. You can tell they're close to single cells. Day 1, they start to either aggregate or divide and grow. By day 3 and then day 6, you can see clear spheroids. And this is the average area of the spheroid over time and increases from about 200 to 700 microns squared. And this is just the time lapse. And these cells were plated in our vitro prime plasticware, not in the imaging dish. And this is over the course of 7 days. And you can see very clear spheroids here. So this is the 2D to 3D cell culture workflow. It's going to look very similar from thawing, but instead of thawing, we're taking existing dishes, we're removing the media, and I recommend always to wash it once with PBS EDTA. That removes the dead cells, and then replace it with Accutase. You could use something like triple or extensive incubation with PBS EDTA to cause single cell suspension. Collect your cell suspension, and you would add equal volume or more of the 3D iPSC Xeno-Free medium with 1x RocketCell cell viability enhancer. So that's going to help them survive the process of centrifuging and counting. Create a pellet like before with thawing. Combine it with the suspension medium. So now you have your suspension medium with 3x of the viability enhancer, and then you combine that with 2 volumes of the Vitrogel stem, place it in each well, wait 15 minutes, and overlay with media. So if you can do the thawing step, you can do the 2D step. If you do the 2D step, you can certainly do the thawing step. We recommend starting with Accutase. We've had a lot of success starting from single cells. And this is an example you'll see at lower resolution and higher resolution using from 2D source. At day 0, you can see single cells or small clumps. And day 1, the single cells either divide or aggregate. By day 2, you see very clear spheroids. By day 5, you can see about 100 micron spheroids. And this is the average area per image normalized at time 0. And then there's approximately a 3-fold expansion in size over the course of those 5 days. And this is all imaged on the IncuCyte S3. So here's the 3D subculture workflow. So now that you have your cells in 3D, what can you do with them? Number 1, you could subculture them and expand them. So at day 0, they're small single cells. By day 7, spheroids. Now, if at day 6, you're happy with the outcome, process them at day 6. You don't have to wait till day 7. If they're slow growing and they haven't accommodated fully to the system, then let them grow a little bit longer. Here you observe the expansion over 7 days, remove the cell culture media, and apply the Vitrogel organoid recovery solution. We have actually 2 flavors, the cell recovery solution, which is a more gentle, and an organoid recovery solution, which has more capability of dissociating organoids. You would apply a 10 times volume to each well. So since each well is kind of limited in volume, you could put 1.5 ml per 300 in the well, and then put that in a tube and add the 1.5 mils back into the well or back into the tube. We've tried it both ways. There makes no difference. Immediately triturate it gently, once or twice, throw it into the water bath or a bead bath for 5 minutes, and go back and retriturate. So the second trituration fully dissociates the gel. If you skip that, you'll notice some issues when you centrifuge. So once it's fully dissociated, you can do 2 things. You can count them, and you may end up with spheroids and single cells, depending upon which reagent you use. So at first, you may want to just do a 1:2 or 1:3 or 1:4 dilution after you centrifuge it and form the pellet. Or you can do further downstream assays, which we'll talk about. So once you have the pellet, you'll resuspend it just like before in the 3D Xeno-Free suspension media, containing 3 times the amount of enhancer solution, and combine it in the 2:1 ratio as we discussed before. So I just want to point out for the 3D subculture workflow, You can also use enzymatic dissociation. So Accumax is like Accutase, but it's 3 times more concentrated and a more pure form. So you can use it in 2D, but it's really important and it works way better than Accutase in our 3D. So we've combined that after either recovery, the cell recovery, or the organoid recovery solution, when we want to go back to single cells or very small spheroids. And you could combine both or just use cell-based filtration, size-based filtration to aid in uniform input. So if these are 1 or 200 microns and you put it through a 40 micron, many of them will get stuck. But if it's enzymatically dissociated, you'll get single cells going through, and any stubborn spheroids that haven't dissociated fully will get caught up. So we've tried all of those to improve uniform input, and this is really important. If you have non-uniform input, so if you have single cells and 100 micron spheroids together, the next level or the secondary cultures will look non-uniform. So please keep that in mind. We're going to go on to some quantitative assays used with the 3D iPSC growth using our kit. We're going to talk about Alamar Blue versus automated cell counting. First, I'll show you the cell counts. So the purple is one cell line and the green is another cell line. These are both iPS cells made with episomal vectors, so they should be not genetically modified. Even though they're very similar, the top one's made from fibroblasts, the other one is from CD34 hematopoietic stem cells, and the growth rate in the purple is a little bit slower. The green is a little bit longer. And you'll notice we took it out to day 11. So we tried to exhaust the system and see how far we can go and how much we could get the cells to divide. So this is about 10-fold after about 11 days. When we compare that to the Alamar Blue absorbance, the pattern is very similar around day 8. What we think we're seeing here around day 11, at least with the green line, is some type of saturation. And again, there's not much difference. So we think we've saturated the Alamar Blue absorbance. But then again, we're only recommending a 7-day work cycle. So you would be carrying it out a day before day 8. So vital dye staining is pretty common in labs. I'm going to talk about something called calcine AM, which is a green vital dye taken up into cells, and DAPI, which is a blue dye, which will be taken up into dead cells. This is a wash protocol, which means you have to wash out the reagent, but it does give you some functional imaging data. So here we're using the red fluorescent protein line and looking at the green fluorescence using a confocal microscope. In this, we're using the 96-well 3D culture and imaging plate. And this is a 3D render, and you can see the maximum projection on this side, but you can appreciate the large green spheroids with smaller dead cells surrounding those. Secondly, we have our own vital dye staining kit. It's called Cyto3D. The advantage here, it's a no-wash protocol. So you put the reagent in, wait the allotted time, and do your imaging. And the staining with the red color is very bright as compared to the red fluorescent protein. So even though we're using a line that has red fluorescent protein, the vital dyes are much brighter, 10 times brighter. So you're able to differentiate based on brightness when you use a confocal microscope, and you can see green and some red, some yellow overlap, which you could discern using this 3D imaging. And now, instead of doing the green and red, we're doing a depth shading to show that we can measure in this well at least 600 microns in depth. And with the tiling function, you're looking at about 1.4 millimeter by 1.1 millimeter. So you can get a lot of data. You're not limited to using the confocal microscope, and there may be some other machines that can scan these in much quicker or much more high throughput. If you're interested more in the Cyto3D kit, there's a webinar on it, but also some information on our website. So we're going to talk about imaging fluorescently labeled iPS cells, those red fluorescent protein cells you've seen in 2D before, in the Vitrogel stem using this kit. and talk about using wide field microscopy, which is capable. Also, the advantages of using something with confocal microscopy. And in this, I'm using the 24-well dish. And again, there's a QR code if you want to find more information. So for the wide field, we're using a Kian system, and you can see the spheroids. They look kind of piled up on each other, and that's because the wide field system doesn't really give you that 3D look or that capability. There's some functionality, but not like confocal. So using a similar well, we took some pictures with the confocal and then render this so you can see that there's many layers. So this is 500 microns, tiled 800 by 900 micron field. And the fluorescence of the red fluorescence is imminently visible in all those layers. So we can visualize fluorescent proteins. How about doing some antibody staining? So live staining is really important. There's some protocols where you do live staining and select cells. In imaging is a little bit different, but you can take this to mean that you can do this for any other protocol, even like flow cytometry. Live EPCAM staining of the surface of iPSCs grown in the Vitrogel stem hydrogel in our kit and using confocal microscopy. And here we're using the 96-well 3D culture imaging plate. And the red color is pseudo-colored in yellow here. And you can see the cell surface staining, and that's rendered in a 300 micron depth in a field of 800 by 900 microns. So live staining is permissible and capable in this gel. And you could continue to use your regular system where you fix cells. So if you can't do the immunostaining the day you need to do them, you can fix it with paraformaldehyde in the same way you do 2D. We just recommend a longer incubation, but you can use directly labeled antibodies. And here we're going to show a Herx dye. So this is a blue dye that permeates live or dead cells, will stain all cells. And this is done in the 3D hydrogel. Antibodies against Lin-28 coupled to a green, Alexa 488. Potocalyxin is similar to Tra-160, so that labels the cell surface. And then also Nanog, which labels the nucleus in far red, or Alexa 647. Similarly, you can use indirect immunofluorescence techniques. So here, the difference is we're going to amplify our signal. It'll look stronger, but what's more important is this requires more washing steps, whereas directly labeled will not use secondary steps. So what we're saying here is the hydrogel and using this 3D imaging and culture plate is very permissible to continue the same almost exact protocols you use in 2D. Again, Herx dye will stain the nuclei, Tra-160 will stain the cell surface, Lin-28 will stain the cytoplasm, and then OCT4 will stain the nuclei. So this system allows you to do all those assays. You really don't have to change very much. There's some small modifications, but nothing significant. And really, iPSCs are just a launching point. We can discover a lot of things about iPS cells in 3D, but you can differentiate these to many different outcomes. Liver, so we're working on iPSC-derived liver organoids, and this is with the red fluorescent protein line. Similarly, you can work on creating intestinal organoids. Again, this is iPSC-derived intestinal organoids, and I'll show you more data about deriving neural lineage cells in the next slide. And if you need more information about the Vitrogel stem, you can scan this QR code. And here we're going to look at the differentiation of iPS spheroids into something called floor plate neuroepithelium. So this is the first step in that two-step protocol that I mentioned before to get cells of the neural lineage. This is kind of like a neural stem cell, but a pre-neural stem cell. And we're going to do that in 3D using Vitrogel stem. So we talked about recovering cells from 3D. So I grew the iPS cells for 7 days, recovered them using the cell recovery solution. So that leaves them in small spheroids. placing them back in Vitrogel stem, and you could use the imaging dish or a regular plasticware. So after 3 days, you'll start to see some of the larger input spheroids increase in size, and some of them will aggregate together and form these organoid-like constructs. And by day 9, you can see tremendous amount of growth. And at higher magnification, you can see many folds resembling these neural tubes. And this is our little buddy for this World Cup, where we got one that looks like a little bear kicking a soccer ball. So you never know what you're going to get in your cultures. But similar ones were taken and stained with 2 steps, anti-FOXA2 with a secondary antibody for green, Alexa 48, Netrin, and Alexa 594. And this is a chicken antibody. This is a rabbit antibody, and this is an anti-NKX 2.2 mouse antibody with Alexa 647. And these are imaged on confocal microscopy. So this is a really good starting point to generate other neural tissue. And here we're looking at the differentiation of those floor plates recovered again into a different hydrogel called Vitrogel neuron. And if you want more information on that, that's a QR code. So we isolated those floor plate spheroids, put them into Vitrogel neuron, and matured them in our neural stem cell medium using our neural stem cell supplement, but using something called the neuron maturation kit cytokines, which are coming out really soon. They're developed for 14 days, and you could see the purple phosphorylated neurofilament network is extensive. And then this overlaps with the choline acetyltransferase, so this highlights cholinergic neurons in green, and any overlap is going to show up in white. So the key takeaways. This is a Xeno-Free 3D iPSC culture. It provides a physiological relevant foundation to develop organoids. So we saw the neural organoids, modeling of diseases. If we took those cells, the cholinergic neurons, and started doing assays on them for Alzheimer's disease or injury, we could use that. And then translation on clinical research as compared to 2D systems. So we couldn't connect all of those neurons in 2D as we did in 3D. The RocketCell 3D iPS Xeno-Free Complete Growth Kit is an optimized all-in-one solution. I want to also include the next point where we include those dishes to really have an end-to-end all-in-one solution where these dishes can take you to high throughput, production, or routine assays with very minimal changes in your protocol. And these are scalable and compatible with standard high throughput screening 3D imaging platforms. So these are the main takeaways. I also want to point out some of the other products that I used in developing the neural stem cells that are available. So this is our just iPS Xeno-Free growth medium that is available separately from our hydrogel. The Xeno-Free NSC growth medium. So our RocketCell platform has that. Right now it's for neural stem cells, and that comes with EGF and FGF. And we have many, many growth factors that have been vetted extremely well with all of our researchers in-house for both advanced 3D and 2D cell culture. And our organoid recovery solution is available separately. It's also companion product to the cell recovery solution. We have some banked webinars, so if you're very motivated to see other ones after you discover this iPS one today, there's a breast cancer model. There's ones that look generally into organoids and the Cyto3D live dead assay kit, and a cell invasion webinar. And you could access those using this QR code. I don't want to plug myself too much, but there's going to be an upcoming webinar in March, robust and reproducible expansion of human mesenchymal stem cells using RocketCell's HMSC Xeno-Free Complete Medium. So it's similar in nature than today, but we're going to use mesenchymal stem cells. And now I want to thank you for your attention, and we can have some questions from the audience.
Hey, thank you, Rick. That's a great presentation. We do have some questions. I think one was we have a lot more. The presentation mentioned that dissociated iPSCs often lose viability when seeded as single cells. How does the optimized 3D enhancer included in the kit overcome this challenge to ensure high post-passaging viability? And how does it compare to TZV or Y compound?
That's a good question. So in the literature in 2D, we used to be able to passage cells from feeder layers using different techniques, cutting them up, and no one was really able to get single cells. And that was important for cloning, especially with mice. So then some discoveries on using ROCK inhibitor, either thiazavivin or Y27632, was fundamental in being able to plate iPS cells or embryonic stem cells as single cells and to be used in high throughput format. In 3D, those reagents do work, but they're not optimal. So what we did is went back to the drawing board, looked at all of our choices, and started testing and assaying. And we came up with a reagent that looks not only at ROCK, but other pathways that would help cells survive. And we combined those into a reagent that's extremely powerful. So technically, you could use it in 2D, but I warn you, you'll have to dilute your cells way more than you're doing right now with those other reagents.
Thank you for that. I have a question for also another one for thanking you, Rick, for a great deck. They said, aside from neural, intestinal, and liver organoid, does TheWell offer cardioid workflows?
So that's a great question. Right now, we're working on that, and there are some major differences of doing it in 2D and 3D. There are published manuscripts using the 3D format for cardiomyocytes, but they're slightly different than our static model. So we are working on that very closely. Thanks.
Thank you, Rick. What are recommended seeding densities to achieve maximum expansion efficiency without compromising pluripotency?
So what's great about our system is it's really tuned to maintain pluripotency and viability with the enhancer. So we recommend in the 24-well format, I mean, that's a lot of the times we'll do assays there, to go no lower than 50,000 cells per well and no more than 150,000 cells per well. So let's go on the higher end. What will happen is you'll get aggregation of cells. So that's not horrible, but it could lead to inconsistencies. And that's where, you could have, and I think everyone could see this picture, and that's our red fluorescent protein. You can see this little small sphere and a much bigger sphere. And so if you initiate cultures in a non-homogeneous way, some outcomes are kind of strange because the larger ones may polarize over time. And that's not anything we could stop in any format because they just get too big and maybe the middle will start to die or differentiate almost like an embryo. And that's the point we make of the advantage of using it. So what I would say is take your cell line and try the 100,000 cells. And if you like what you see, continue with that. If not, go lower. And you can even try one cell and see if you're able to clone. We've had some situations where I've input very small amount in our 2D format and get the cells growing. So I know the media is robust enough, but in 3D, I would stick no less than 50,000 at first.
Okay, thank you. For the attendees that have their hands up and want to ask questions, there is a Q&A button on top. You should see that. You can write your questions there if you're not aware. That would help us to know what questions you have if you're raising your hand. Okay. We do have next question is the weekend free workflow allows for alternate day feeding. Does this schedule change significantly if I am expanding HESCs versus iPSCs, or is the protocol standardized for both pluripotent stem cell types?
That's a great question. So if I knew which cell line you were growing and I grew it before and there's literature on it, I would be able to give you an answer. So each individual cell, and there's no way of predicting it, will grow at a different rate from 2D to 3D. And you'll have to determine that empirically. The most important thing is the weekend free, 2D or 3D, you put the lowest amount of cells possible. And so in 2D, it's really clear. In a 6-well dish, I'll put in around 100,000 cells, maybe 50,000 cells. In 3D, you could put the 100,000 cells on a Thursday, double up on your media over the weekend, and everything will be fine on Monday. And go ahead and do your alternate day feeding. You wouldn't want to have your weekend free at the end of your 7 days. You would have to put a lot more media. Now, even though our imaging dish does allow you to put more media than a regular standard culture plate, I would feel uncomfortable advising you that way. So again, start off with the low amount, and it will support it, both embryonic stem cells and iPS cells.
Thank you. We do have a few more questions. And compared to a traditional method of thawing into 2D first, what are the expected recovery rates at day 0 attachment efficiencies for this direct to 3D approach?
So the short-winded answer is, I don't know. And the reason is, I don't know how well you've prepared your frozen cells, but we recommend, if you have the ability to thaw acutase or triple dissociated cells. So if your input is single cells, the recovery is best. And the reason is, if you start off with small clumps from 2D, so there's a technique to use PBS EDTA and not go fully and then triturate that and freeze small clumps. Those will survive better in 2D. The reason is, when you triturate 10 times gently, unless you're using a very wide bore pipette tip or a 5 mil glass pipette, that trituration will introduce a shear force. So if you're going to do the 3D thaw, firstly, make sure you're using single cells, and the efficiency is great. It's wonderful.
Perfect. Thank you. The presentation showed that iPSC is expanding and then differentiating into FPNE organoids and mature neurons. Is the RocketCell medium designed purely for expansion, or can it be used as a basal starting point for these specific differentiation protocols?
So that's a good point. Our basal medium, before it's supplemented, probably can be used for many different purposes. But what we're doing is designing a kit that would support things more optimally, which means, to go to the floor plate, we're going to be using our neural stem cell basal medium, our neural stem cell supplement, but create a tailored floor plate cytokine addition. So I don't know if that answers the question, but in the similar manner, use a very similar input for the neural stem cell or floor plate to mature neurons. And then in that case, we would change our hydrogel in a kit that would support the maturation of neurons.
Thank you. I see we have 3 more questions left, so I think we'll run those through. The physical properties of a hydrogel can influence iPSCs, self-renewal, and differentiation. What are mechanical properties of Gel stem hydrogel, and how do they compare to other common 3D matrices like Matrigel or synthetic hydrogels?
Okay, so I'm going to answer that using another hydrogel and then circle back to Matrigel. So our Vitrogel stem is tuned for iPS cell or embryonic stem cell growth. That doesn't mean other cells can't grow in it. However, when we shift to maturation of neurons, we shift to Vitrogel neuron, which is slightly softer, but more importantly, the functional groups are different. So not only are you talking about the consistency or the tension that the gel introduces, but the Vitrogel stem is tuned for iPS cells. So Matrigel, which is an animal product, has probably thousands of other chemicals in it, or not small molecules, but proteins, ECM. So it's very hard for me to predict, but those hydrogels would be very different. I think if the amount of Matrigel you would need to get a 3D hydrogel would be much more, and I'm not sure how iPS cells would grow in a 3D Matrigel matrix that would be, being able to be set up in the same manner that we have. Other synthetic ones, again, I don't know how they compare, but again, these our iPS and our neuron ones were specifically tuned to known tensions or known rheology based on research. So they're very reproducible. I hope that helps.
Yeah, I think that's a great answer. For the researchers looking to scale up production, example in spinner flasks for bioreactors, is the VGL stem hydrogel compatible with these systems, or is it primarily designed for static culture?
So that's a huge challenge for us. And so what you're seeing now is kind of the entryway into that. So if you're interested in scaling up into more of a liquid-based assay, we do have protocols for it where we dilute the static system a few times with media and some other components to help survive the shear force of these bioreactors. So yes, our media is compatible, the hydrogel is compatible, and the big question mark is your bioreactor safe for iPS cells? And if it is, and you know how to use it, then our system will suffice for it.
Thank you so much for the answer. Last question here. I think that the plate features a cover glass bottom. I believe it's a RocketCell. Is it compatible with advanced techniques like high NA oil immersion objectives, confocal, and multi-photon microscopy?
Okay, so number one, I wish I had all of those machines in-house. We do have a confocal microscope, so it is very compatible with confocal microscope. Again, it's compatible with wide field microscopy. If your confocal microscope has longer working distance objectives, that's the best. It is a cover slip bottom, but it's raised ever so slightly above the lip. More dishes have longer working distances. We have the shortest possible, aside from having it rest right onto the surface, which would be deleterious to the glass. We felt it was going to break. We also have used our dish in other imaging platforms. So I did show you some information about using it on an IncuSite, and that has some fluorescence capabilities. It does work in that platform, and we're getting it tuned perfectly for that one. And we have tried it in other imaging platforms with high throughput success, and it works just wonderfully.
Thank you. Thank you. I think that was the last question for our segment. And thank you, Dr. Cohen, and everyone for continued support of this webinar. Again, we have an upcoming webinar on March 18th focused on MSC culture. Please connect with us if you have any questions about our product or any questions about this webinar to Dr. Cohen. His contact, his information should be there. If not, reach out to us and we'll connect with you with our support team. And visit our website for any more about our hydrogel products and various 3D cell culture applications, protocols, scientific resources, and publications and future webinars. If you have any more questions, please email support@thewellbio.com. Have a great day, everyone. Thank you again for joining our webinar. Thank you again, Dr. Cohen, for excellent webinar there.
Thanks, everyone. Have a great day.
Thank you.